Hello. In this video, I want to talk to you about the very earliest stages in the development of the heart. And essentially what we're going to be looking at is how do we go from a flat sheet of cells to a tube, and then the earliest stages of how that tube starts to organise itself in preparation for looping and formation of the adult heart chambers. Now, it is sensible, first of all, just to very quickly revise the stages of early embryonic development, focusing on structures such as the embryonic disc and gastrulation. So I've got a summary figure here on the left-hand side, and you should remember these from earlier on in the course. Essentially, what we have is a layer of cells like the interface between two bubbles, those bubbles being the yolk sac beneath and the amniotic sac above. In the early stages, this interface, this layer of cells, has got two separate layers, but we need to go from 2 layers to 3, and that is the process, of course, known as gastrulation. If we were to look down upon the layer of cells that will become the body from above, we see a structure known as the embryonic disc. So in the pale blue here, we're looking at the embryonic disc here. And the layer that we're looking down upon is the ectoderm, the outermost portion of the embryo, which you should recall goes on to form the skin and central nervous system. Furthermore, when we look down upon this embryonic disc, we can also see a structure known as the primitive streak. And if we look at it from directly above in this figure here, we can see the primitive streak in a little bit more detail. Now, the primitive streak is very, very important during development, since this is the site at which these outermost cells, which as we said will become ectoderm, invaginate to form the mesoderm. So it is at the primitive streak that gastrulation occurs, and we end up going from a 2-layered disc to a 3-layered disc with outer ectoderm, inner endoderm in yellow, and sandwiched between the two in red, the mesoderm. And it is the mesoderm that we're going to be focusing upon because the heart and much of the cardiovascular system develops from the mesodermal layer. So let's do a little bit of drawing and try and understand where the heart comes from. I'm going to start off by drawing a view looking down on the embryonic disc, just as we saw in the diagram. So here we're looking down on the embryonic disc here. And what I can do is I can add on the primitive streak just there and the primitive node just at that point. Now, we said that the heart develops from mesoderm. Mesoderm, as we said, is sandwiched between ectoderm and endoderm. And the mesoderm undergoes differentiation depending upon which structures it is going to ultimately go on to form. Of course, a large portion of the mesoderm goes on to form the somites, which contribute to the musculoskeletal system as well as the skin. But we're going to be considering a specialised region of mesoderm which forms a sort of horseshoe-shaped structure just here. So I'm superimposing upon this upper layer of the embryonic disc the location of the so-called heart field, okay? So this is our heart field, which contains the mesodermal cells which are going to go to form the heart and many of the major blood vessels. So this is our early heart field, a specialised region of mesoderm. Now, even at this early stage in development, we can see something intriguing and in fact highly clinically relevant. If you speak to most people, they will give you an account of the heart, something along the lines of it being primarily on the left-hand side. And of course, anatomically, we know that a large portion of the left ventricle does indeed protrude into the left half of the chest. However, when you look at the development of the heart, you notice that the heart field is in fact a bilaterally symmetrical structure. So the heart even from the earliest stages of development, has arisen from both the left and right hand sides of the embryo. The relevance of this is that in a myocardial infarction or in angina, patients can experience pain on either the left or the right halves of their bodies. Of course, classically, we're talking about radiation into the left arm, but it is most definitely seen that in some patients, their pain radiates over to the right-hand side. And that is down to the fact that the heart has originally developed as a bilaterally symmetrical structure. So what we're going to do is we're going to draw a cross-section through the embryonic disc at this level here. Okay? And we're going to take a slightly closer look at what's going to happen. So we're going to draw our cross-section down here. And if I this is a very, very simple cartoonish image, but if I'm drawing the cross-section, it'll look something like this. Okay, what I've just drawn there is the ectoderm with the neural folds raising up, and this is the neural tube in the process of development. For completeness, we can pop the notochord just down there beneath the neural tube. What we then see is that we cut through our heart-filled cells at 2 points, because this is a horseshoe-shaped structure, isn't it? So we have heart-filled there and there. And you note that they've formed two tubes, okay? They've formed two hollow structures, two tubes. And what is in fact going to happen is that these two tubes from each heart field are going to come together in the midline, such that the next stage is that the heart field from the left and the right hand sides comes together to form a single tube in the midline. And the stage at which this occurs is at approximately the 3-week stage. Okay, so by around 3 weeks, we have got a single tube in the midline. And this tube is going to then go on to form the entirety of the heart and the great vessels. So what we're going to do next is look at this process in a little bit more detail and look at the specific regions of this primitive heart tube. So, thinking about the development of the heart, it's a tremendously complex process. We've got to go from what is a simple tube to something that looks as complex and functions as complexly as the adult heart. And this is really quite a remarkable process. The stage that we're going to be looking at the heart tube is when it is about 22 days old. All right, so this is immediately after the heart tube has formed. And at about this age, this primitive heart tube is in fact beating, because it is from this age that diffusion is no longer sufficient to provide nutrients to the body. We need a cardiovascular system. So let's draw this primitive heart tube to see what it looks like. I shall draw the outline first and then I'll talk you through it. Okay, so I'm drawing some vessels here. These vessels are connected to one swelling, and that swelling is connected to a second swelling, and then that second swelling is connected to some additional vessels. Okay, so what I've drawn there is the primitive heart tube. And it's interesting to note that this primitive heart that I have drawn, this actually resembles the heart of a fish. So the heart of a fish is a very simple tube, not dissimilar from that seen in a human embryo. But we know that in the human heart, this tube has got to be partitioned and we've got to form 4 chambers. So there's got to be considerable remodeling taking place. Let's label this. Blood is coming in from this direction. So the venous end of this heart tube is down at the bottom and the arterial end is up at the top. So let's just label the arterial end with a red A and the venous end with a blue V. Now, please, at this stage, just remember that there isn't segregation between oxygenated and deoxygenated blood. I'm simply using red and blue since they offer a nice contrast against each other. Down, and you can see as well that separating the various parts of this primitive heart tube are some narrowings. So there is a narrowing here, there is a narrowing here, and there is a narrowing here. Okay? So we already have some differentiation of the different parts of the heart tube, and that is visible by the fact that they are relatively narrow at 3 points. The first swelling that we have is the primitive atrium. So this is the primitive atrium, okay? And there is only one at this stage. We don't have a right and a left atrium at this stage. We just have a single primitive atrium. Sitting above the primitive atrium towards the arterial end is the primitive ventricle. Now, as you know, atria are connected to veins, whilst ventricles are connected to arteries. The arterial end of this primitive heart tube, forgetting at the moment about it splitting into two, but the arterial end is known as the truncus arteriosus. And the venous end, that is what's going to become the veins draining into the atrium, is known as the sinus venosus. So what we can see now is something which is starting to bear some correlation to the adult condition. We have an atrium and a ventricle. The atrium is receiving blood from the sinus venosus, and the sinus venosus is going to become our great veins, whilst the ventricle distributes blood through the truncus arteriosus, and the truncus arteriosus is ultimately going to become our great arteries. Now, to summarise the entire process, we've got to add on some additional features and consider some additional events. The first thing I want you to note, with the exception of the inferior vena cava, in the adult heart, all of the vessels are attaching to the superior aspect of the heart, whereas in the embryonic heart, the veins are at the bottom and the arteries are at the top. And what this tells us is that during the development of the heart, there must be folding of this tube or looping of this tube. So this tube must loop during development in order to bring most of the vessels up at the top. The second thing that we need to appreciate is that we know that in the adult heart, not only are the arteries and veins segregated, but furthermore, the veins going into the right atrium are separated from the veins going to the left atrium, and the arteries coming from the right ventricle are separated from the arteries coming from the left ventricle. So there must be atrial and ventricular septa separating the ventricle into two halves, as well as a mechanism to separate the veins and arteries. So we can add a further line like this just to demonstrate that we need to separate this primitive heart tube into a number of distinct regions. So you can imagine that, of course, this region might become the left ventricle, that the right ventricle, that the left atrium, that the right atrium, this the vena cava, etc. So we've seen how we can take this primitive heart tube and subdivide it. Now, why do we need to subdivide this primitive heart tube? Why would it not be adequate for us to just scale up this fish-like heart to air-breathing mammals like us? Well, that's the important phrase, the fact that we breathe air. And in order to breathe air, you need to have two vascular circuits. A pulmonary circuit, which is going to the lungs, and a systemic circuit, which is going to the rest of the body. So we need to septate this primitive heart in order to provide us with those two circuits. An additional and final point before we move on is that you'll appreciate that this process of septation and folding that we're going to describe in more detail later on, like all events in embryology, must be highly complex. And like any complex event, it does have a tendency to fail sometimes. So if there is any problem with any of these events of forming septa within the atrioventricles or of folding and looping the heart, this could lead to congenital heart defects. So let's now move on and take a closer look at an important event in the development of the heart, which is this cardiac looping, where we're going to go from a simple tube to something a little more complex. As with the previous slide, I've included an image of the adult heart, just so that we can refer to it as we're considering the development. The first thing I want to do is I want to draw for you a view of the primitive heart tube, but more from a side-on view, okay? So we're going to look at this from the side. So we can see here we have got the truncus arteriosus with a couple of vessels coming off. Then we've got our primitive ventricle, our primitive atrium, and then finally our sinus venosus with our various veins plumbing in. Okay. Now, the event that's going to happen, and remember we're looking at this from side on, is that effectively, I mean it's a little more complex than this, but effectively the atrium is going to elevate, is going to rise up and find itself sitting behind the ventricle. Okay? So we're looking at this from the side in this little sketch I just did, and the process I'm going to show you is that the atrium is going to bend backwards and upwards, so that it finds itself behind the ventricle. So let's draw that and try to depict what the thing will look like from the side if the atrium elevates and finds itself behind the ventricle. So once again, we can draw our truncus arteriosus here, going into our primitive ventricle. Okay. And because the atrium has gone behind the ventricle, we need to draw it like this. And here, at this point, is the atrioventricular junction. So this point here corresponds to this point here that I drew on the left-hand side. We'll label this is the ventricle. And then here is our primitive atrium with our sinus venosus vessels attached. And then that goes on to form the bulge of the primitive atrium, which is now sitting posteriorly. Okay. So what we've got now is something that's starting maybe to look a little bit like an adult heart, a little bit more like an adult heart, because we do know, don't we, that in the adult heart, the ventricles are sitting primarily on the anterior surface. And that's the situation that we've got here. So if I just remind you that this is anterior and that this is posterior, you can appreciate that we are now approaching a situation where the ventricles are forming the bulk of the anterior surface of the heart. Now, this narrow waist-like atrioventricular junction is a very important region of the developing heart because it is at this point where the valves are going to form. Okay, so this is the AV, the atrioventricular canal or atrioventricular junction, and the atrioventricular valves are going to develop in here. Okay, so we can start to see something that's resembling an adult heart. It looks even more like an adult heart, however, when we look at this from the front, okay? So we are now going to look at this structure from the front. So this is as if this was my eye, okay? And it is the view I would have if I was looking at this heart from the front. And if I show you what that view looks like, it will look relatively reminiscent of an adult heart. So here is the truncus arteriosus with its two branches. And here is the primitive ventricle there, okay? The atrium, remember, has gone behind the primitive ventricle. So here are the veins of the sinus venosus. And we can see the rest of the primitive atrium if we depict that as dotted lines just here. And it just protrudes like that and like that. Okay? So now we can see something that's starting to look like an adult heart. All right? To recap, here's the truncus arteriosus on this anterior view. Here is the sinus venosus. This is the primitive ventricle sitting relatively anteriorly, as we said in the side view. And we can see the atrium sitting behind the primitive ventricle with these two small protuberances visible to the sides. Now, this view here, I think, is really helpful. You know, I think that appreciating that the atria have gone behind the ventricles helps us to orientate ourselves in the adult heart. If we look at the adult heart, we see that we can tell if we're looking at the front of an adult heart because we should be able to see these structures here, the auricles, okay? The auricles, the right auricle and the left auricle. And I kind of think of the auricles, they're part of the atria. I think of the auricles as kind of coming from the back of the heart and embracing the ventricles. And isn't that similar to the situation that we drew here, where the atria have gone behind the atrium has gone behind the ventricle, and these portions of the atrium sticking up will become the auricles. So that's the first thing I want to talk about with respect to cardiac looping. The second aspect of cardiac looping is going to help us understand some features of the pericardium known as the pericardial sinuses. So what we've got to do now is draw our primitive heart tube but add on the pericardium. So let's draw on a very simple primitive heart tube. I'm not actually going to put the atrium or any of the other swellings on. I'm just going to draw it as a very simple tube. Whoops. So just a very simple tube. Like this. Okay, so here's our primitive heart tube. The venous end is at the bottom, so blood is coming up and entering it in this direction. And the arterial end is up at the top, so blood is leaving in that direction. Now this primitive heart tube is embedded, if you like, it has invaginated into the pericardium. And I'm going to depict the pericardium in green. So the pericardium has two layers. It has the visceral layer, which is in contact with the heart directly, literally stuck to the heart. And then it has the parietal layer, which is sitting more superficially. And in between those two, we have pericardial fluid. So this space is filled with pericardial fluid. I'll draw that on the other side as well. So we have visceral pericardium in direct contact with the heart. And then we have the parietal layer sitting more superficially. So that's how the heart and the pericardium are related to one another. You could alternatively think about this arrangement as resembling a hot dog in a hot dog bun, okay, where the heart tube is the hot dog and the pericardium is the bun. What's going to happen next is, of course, this heart tube is going to lengthen and is going to loop on itself, all right? Now, the loop, of course, is complex, but conceptually, it can just be thought of as bending this thing into a U shape. So we're going to bend this into a U shape in this direction here. And when we go through that transformation, we get to the point where it looks a bit like this. So here is our truncus arteriosus. Here is our now U-shaped heart tube because it has looped. And then here's our sinus venosus here. It's all still enclosed within pericardium. So we can draw our pericardium on like this. Okay, remembering that I think of the pericardium, as I said, as like a hot dog bun with the heart tube forming the hot dog sausage itself. So here's the visceral layer of the pericardium and then the parietal layer there. So the heart tube has folded, taking the pericardium with it. Still, this is the arterial end and this is the venous end. Now, the consequences of this folding and its relation to the pericardium are anatomically very important and very interesting. The first thing that you will note is that the arteries and the veins are clearly distinct, and there is clearly a gap between them, okay? So there is clearly a gap between the arteries and the veins. So this gap here between the arteries and the veins, where the two come together following the folding, is known as the transverse pericardial sinus. Okay? So this gap between the arteries and the veins, which occurs due to the folding of the heart tube, is known as the transverse sinus. The transverse pericardial sinus. Okay? And we can see the transverse sinus in the adult heart down here. So down here, a finger has been inserted through the transverse sinus, and that finger sits between the aortum pulmonary artery and the veins. Okay? So the transverse sinus separates the arteries from the veins, and you'll note that the arteries sit relatively anterior. Now, the clinical significance of the transverse sinus is there may be a time where you might need to clamp the arteries. For example, if you're setting someone up on bypass or doing other procedures. And so, therefore, you need a plane that you can pass your clamp through, and that would be the transverse sinus. There's a second sinus we need to appreciate. And what you've got to imagine here is that you could enter the pericardial cavity at this point and then pass your hand behind the heart tube, okay? So this one's a little more difficult to appreciate from the folded tube description, but still imagine that you could pass your hand into the pericardial cavity, but then put your finger behind the heart tube itself. This space is known as the oblique pericardial sinus, the oblique sinus. And in the adult, it's much easier to appreciate because in the adult, here is an image of the pericardium with the bulk of the heart removed. But you can appreciate that there is this space here that's sitting behind where the heart would be. And that is the oblique sinus. And the top of the oblique sinus is in fact up here amongst the pulmonary veins. So simply speaking, the oblique sinus is a space within the pericardial cavity behind the heart. Clinically, the oblique sinus is less significant than the transverse sinus. However, you do need to appreciate it because in performing cardiac surgery, you may have placed swabs in this space. And it's always worth checking the oblique sinus to make sure you didn't leave anything in there. So this is cardiac looping and the relevance of that to the transverse and oblique cardiac sinuses. We're almost done for this section now, but I want to finish by talking to you in a little bit more detail about the atria. Okay? And we're going to particularly focus on the derivatives of certain parts of the atria with respect to the adult atria and also parts of the conducting system. So let's just draw the lower half of the primitive heart tube. So we've got our primitive ventricle here, we've got our primitive atrium here, and then we've got the veins of the sinus venosus just here. So to label that, we've got ventricle, then we've got atrium just there. Now, it's interesting to note that the adult atrium does not develop from the primitive atrium. Let me rephrase that. The entirety of the adult atrium develops from both the primitive atrium and elements of the sinus venosus. Okay, so the adult atria develop from the primitive atrium and elements of the venous system, the sinus venosus. And the consequence of that in the adult is that when you look inside of the right or left atrium, you note that there are certain areas which are rough and certain areas which are smooth. The rough areas develop from the primitive atrium, whilst the smooth areas develop from the sinus venosus. So let's draw that in a little bit more detail. So if I do just a bit of a higher magnification view of this region, and this could be either atrium, but we're going to focus on the right atrium, and you'll see why that is shortly. Just very simply, very diagrammatically and cartoonishly, here is the primitive atrium, and here are just two of the veins of the sinus venosus. Okay. Now, you know that, of course, the atrium ultimately will become partitioned with the atrial septum. And we said, didn't we, that the adult atrium is formed from a contribution of both the primitive atrium and the sinus venosus itself. The rough portion of the adult atrium is derived from the primitive atrium. So the rough portion, which I'm depicting with this kind of shading, is the portion of the adult atrium derived from the primitive atrium itself. Whilst the smooth portion of the atrium is derived from the sinus venosus. Okay? So these two regions that I've drawn will both go on to become the adult atrium. Okay? So the adult atrium is formed from contributions from both the sinus venosus and the primitive atrium. Now it gets a little bit more interesting than this, because from the smooth portion of the within the smooth portion of the adult atrium, that which is derived from the sinus venosus, we develop the sinoatrial node. So the SA node, the sinoatrial node, important for the rhythm of the heart, is actually derived from the smooth portion of the atrial wall, which comes from the sinus venosus. Okay? Whereas the rest of the atrium, including the auricle, are from this rough portion which develops from the primitive atrium itself. Okay, so that ends this section. Thank you for listening.